All right, so we're going to do our diffusion lab part 2 or procedure 2 today. And we're going to be doing the solutions that I talked about inside of the dialysis tubing, inside of the cups that I mentioned in the last video. But obviously, I'm going to explain it in more detail. So let me kind of walk you through the setup for the demo that I'm doing. And then we're going to collect data together. All right, so as you can see for the agenda, we are doing diffusion lab procedure number 2. The objective is still the same as it will be for this whole unit. Make sure that you are completing your chapter 5 summary and Learn Smart because that is due on Friday. And your cell drawings are also due on Friday from page 64 of your textbook. The only thing that's actually due today is going to be your diffusion lab procedure 2 lab report, which is not very long. But if you get confused along the way, then it does make it a little bit harder to understand. So first off, we're going over this. So if you are in the hospital and you need an IV, you read the label on the IV bag, which lists all of the solutes that it's dissolved in a liquid. That's actually like the bag, right? So why is it important for an IV solution to have salts in it? It's because your body has salts in it. And if you put something into your body that does not match your body's salt level, your body's, if there's too much water in it, or if there's too much salt in it, the water is going to leave or enter your cells and cause them either to shrivel up and die or to burst and die. So that's why the amount of salt in the IV fluid has to match the amount of salt level concentration in your own cells. So if you are given pure water in an IV, that means the water would continuously go into your cell because your cell has a semi-permeable membrane, and it's going to flow from high water potential to low water potential. And your cells are going to have lower water potential because they have salts and sugars dissolved in the liquid of your cytoplasm, whereas pure water is just water, so it has the highest water potential. And so when you go from the higher water potential to the lower water potential, they're never going to equilibrate because your cells always have solutes in them. So water is going to keep going into your cell, into your cell, into your cell until your cell, as an animal cell with no cell walls, is going to burst and die. Okay, so if you have pure water in an IV, it can kill somebody really quickly. How would you determine the best concentration of solutes to give a patient in need of fluids before you introduce the fluids into the patient's body? You can test your own salinity by drawing a little bit of blood to check what their actual concentration of salt, concentration of sugar, and concentration of any other chemicals in their blood currently is. So we're going to be doing something based on cells, but we're not going to be actually using cells because these are on a microscopic scale. So we're going to be using dialysis tubing. And this is actually, I did a food coloring one to show you guys what it is. But basically, it's a tubing that's like a clear kind of plastic baggie, but it's actually a big tube. And I've tied one end of it off to close it. And then I put the solution inside of it, put the food dye in so you can actually see it. Left a little bit of room just in case we want to see if water is going to enter or leave. And then I tied the other end to seal it off. And this is basically going to simulate a cell for us because the whole outside plasticky part is actually a semi-permeable membrane. So things can move across, but not all things can move across. Things that are too big or too charged won't move across. So we're going to go ahead and walk through the setup and explanation of the lab. So like I said before, our options are pure water, 1 molar sucrose, which is a sugar solution, 1 molar NaCl, which is a salt solution, 1 molar starch solution, and we're going to have our dialysis tubing, which represents our cellular membrane. We're going to have cups to put them in together with as our setup, and we have a balance because we're going to be comparing masses. So how can you use the weights of the filled cell models to determine the rate and direction of diffusion? Well, if the cell, the tubing, if the cell weighs more at the end than it did in the beginning, that means something went into it that caused it to weigh more, right? The contents increased, right? So you're going to think about osmosis in that respect. And if you weigh the cell before and the cell after actually weighs lighter than before, that means something left the cell. And so we're going to think about osmosis and what is leaving the cell, and the contents have decreased, so it weighs less. So diagram the flow of water or osmosis in your reaction, label inside, outside. We're going to actually draw these out for 2 separate cups. Okay. The previous page actually asks right now, what would be an appropriate control for the procedure you just described in order to make sure that we don't have any mistakes going on and that we just have, for example, that water movement is not based on mistakes in the tubing having holes, the cup having something extra permeable in it. You would actually do one with water on the inside and water on the outside to make sure that if the mass does not change, then your other cups with materials are not affected, and then you can take data like normal. But if you logically understand that, that would be the control, then we're going to move on and skip actually doing that part. So I want you to draw one diagram like this. Some of you have already done it on your previous notes, which is good, but for this diffusion procedure 2 lab report, I'm going to want you to draw one diagram like this and one like the next page. Okay, so you can do them on 2 separate pages if that's easier for you. So one is a cup or beaker, and the solution inside of the beaker is going to be pure water. Okay, and we're in that water, we're going to put some iodine because it's a starch indicator. So if the starch and the iodine touch, wherever they touch, it's going to turn black immediately. And we know that iodine is originally this kind of reddish brownish color. So you can kind of see through the neck right here of the bottle. It's a reddish brownish color. But once it touches starch, which is originally a white color, it turns black, like pitch black blue. Okay. Inside of the dialysis bag, we're going to actually have, inside of the cell, we're going to have our 1 molar starch solution. And obviously our bag is representing our cell membrane. So I want you to draw out this diagram, so you can pause if you need to do so. On another diagram, for our second setup that we're doing, right, we're not doing the control, so we're not doing the third, but for the second setup that we're doing, maybe if you want to do it on another page, we're going to have basically the exact same thing, except with the outside solution, we're going to be using a 1 molar salt solution instead of that pure water. And that is the biggest difference here. And we're going to see if that actually makes any impact on the way that water moves, on the way that starch moves, on the way that iodine moves. Okay, and notice that with the salt solution, we're still having iodine. Okay, because we want to have a starch indicator. So originally the starch is on the inside. So we're going to put the iodine on the outside to see where does the iodine go, or where does the starch go. And we can see which one is moving and which way it moved based on where you can see the black particles that are going to be produced when the starch and the iodine interact. So pause and draw this diagram on a separate page or on the back of the page. Now, under each drawing, you're going to be writing 2 hypotheses. Okay, so the first one under both right now. So this hypothesis is going to go under both, and then this second hypothesis is going to go under both as well. Okay, so you're going to write one for the water cup, and you're going to write one for the saltwater cup now. So you're going to write an if-then hypothesis, predicting what will happen to the movement of solutes. Right, we're talking about the salt, we're talking about the starch. Okay, we're also talking about the iodine. So diffusion hypothesis is assessed by color change. So this is the one we're trying to see when the starch and the iodine touch. Where are they touching? Is it inside of the cell or outside of the cell? And what color is it turning? So the example set up for this hypothesis would be if the starch diffuses across the semi-permeable membrane, then the blank of the beaker will turn blank. Okay. Actually, let's save this. Instead of the beaker, let's change this one to cell. So we're going to talk about, is the inside or the outside of the cell going to turn? What color does starch and iodine react to become? Right. If you need to look it up again, you can. If you want to watch over the video that I just explained in, you can. Okay, so you're writing this under both of the cups, right? Because when we have a starch cell inside of a water cup, and when we have a starch cell inside of a saltwater cup, okay? So if the starch diffuses across the semi-permeable membrane, then the blank side of the cell will turn blank color. The next hypothesis you're writing under both of the cups diagrams that you drew is going to be predicting about the movement of water. Okay, so we want to talk about osmosis hypothesis, and we're going to assess this one by mass, not by color. So for example, if the water diffuses across the semi-permeable membrane, then the tube will weigh more or less. And again, we're going to change this. Instead of saying tube, we're just going to call it a cell, just to be more physiologically correct on what we're trying to represent. Okay, this one's pretty simple. This is the reason why we're doing mass and percent change in mass. Just a caveat though, on this previous one, you don't have to write about starch. You can write about salt, you can write about NaCl, You can write about water. You can write about iodine. But you have to make sure that you are talking about the ones that will actually change color when they interact with each other. So now you should have 2 hypotheses written under both of your diagrams. So technically you should have written 4 in total. So here's the setup that you're not really doing, but I am. So it's basically going to be 2 separate setups of this. And we're going to actually start out and mass our cells from the beginning. So you have your initial masses, and then we'll have a final mass at the end. So let me go ahead and show you how you're setting up your table. So I have the formula over here because you're going to need it. And if you want to, write it down on your page because it is a very useful formula. So percent change in mass is going to be final mass minus initial mass divided by the initial times 100. Okay, so you're setting up your table like this. So you want to label which cup is which. So the first cup we're going to talk about is the inside is the starch and the outside is the salt water. And then cup 2, the inside is the starch and the outside is the pure water. You just want to put your data table under whichever diagram that you actually drew. Okay, it doesn't matter what cup number you have. I just want you to make sure that you have it under the correct diagram that you drew. So the one that has salt water versus the one that has pure water. So you want to have a section for initial mass. You want to have a section for initial color of the tube. And again, I'm going to be adjusting these to say cell. Then you want to have a section that says final mass, final color of cell. And then you want to one last section that gives you room to do your calculations using the initial and final colors, masses of your cell to do percent change in mass. Okay, you want to do the exact same thing for your other cup or your other setup. And you want to have initial mass, initial color of cell, and final mass, final color of cell. And then you're going to be using your final mass and your initial mass to calculate percent change in mass for both. Okay. After you collect that, you're going to be writing out your statements for diffusion and your statements for osmosis, which means you're going to have a conclusion for diffusion for the first cup, a conclusion for osmosis for the first cup. Okay. And then you're going to rinse and repeat, and you're going to do a conclusion for diffusion for the second cup and a conclusion for osmosis for the second cup. So in total, you're writing 4 short paragraphs. Okay. So first of all, you would be saying for cup 1, you want to talk about what happened to the colors. Okay. You want to tell me basically, based on where the color is, which way did a solute move? Across the membrane into the cell or across the membrane out of the cell? Okay. You want to write one sentence knowing how you know that occurred, right? Because you should know where the solute started. You should know where the salt started. You should know where the iodine started. You should know where the starch started, right? And then you're going to write one sentence whether or not your hypothesis was correct that you initially made. Remember that first hypothesis that we made for both? You're going to be telling me if that is correct or not supported, okay? So if your hypothesis has been falsified, you're going to write a new one that actually matches with the results of what you have, right? So maybe the reason your hypothesis was wrong was because you were talking about the wrong solute, or maybe you said the direction it was going to change color in was wrong. Or maybe you said the wrong part of it was going to change color. And then you're going to write one sentence suggesting an additional experiment that you could do that would support your original hypothesis, or you can test your new one. Okay. Similarly, for the osmosis conclusion paragraph, you want to write one sentence that it tells me the calculated percent change in mass that you calculated after the final and initial were taken. You're going to write one sentence stating what the results indicate. So which way did water move and how do you know, right? Which way did water move? Because that's literally what osmosis means, right? The direction that water moves. And then you're going to write one sentence stating how you know, because you should know what the initial weights were, and you want to tell me basically what is changing. Is it increasing or decreasing? And then you're going to write one sentence stating whether your hypothesis is supported. And if it's not, please write a new one that actually follows what the data says. And then you want to suggest one additional experiment that will support your original one, or test your new one if your original one was wrong. So in the end, again, you're writing 4 hypotheses, and then you're going to be writing 4 separate conclusions, one for each of those hypotheses. Okay, so I'm going to actually go over the lab setup with y'all now. So we're going to go over and we're going to actually mass out the initial masses and make the initial observations. So I want you to make your initial observations of the cell color. Pretty obvious, right? They're both white because starch is white, right? Remember, we have the starch solution inside of the cells. And so the color initially is this. Okay, I've left them a little wrinkly because we want to leave it in room in case water goes in. We also want to make it full enough that we can tell when water leaves, so it gets more and more saggy. Okay, so we have our two, and we're going to mask them. So we're going to put on a cup to hold them, and then we're going to click on, and we're going to wait for the scale to turn on and to set itself. And then we want to click 0 at the bottom here. It usually says, let me move my, it usually says 0 or tare, T-A-R-E, and it's going to then make the cup mass 0, so that the only mass that you're calculating is going to be the mass of the tube. And so we're going to put in the tube for the NaCl cup now, the salt solution cup. And that is going to be 10.796. We're going to round to the hundredths, which means that it's going to be about 10.8 since these numbers all seem to round. Oh, it's going to be 10.79, which means our data right here is going to be 10.79 grams. Okay. And then we're going to go and we're going to clear this up. Make sure you hit tare again, or 0. And then we're going to weigh the one that's going into the water cup. And that one weighs 12.50. So we're going to record 12.50 grams. Okay, so you want to write down your initial color of your cells in each of these, and then we will find out the final mass and final color of the cells after we have let them diffuse for a while. So now basically I'm going to be setting up the 2 cups. So one of them, this one right here, says starch inside and then NaCl and iodine on the outside. This one says starch on the inside and then pure water with iodine on the inside. Okay, that means that before I want to put in my little cells with starch, I'm going to want to add iodine to the solutions on the outside. And as you can see, the iodine color is this brownish reddish color. When you add it to water, it gets diluted a lot, obviously. We put about 2 full squirts into each one. And we're going to make sure that it has time to kind of spread itself out. And then we're going to put our cells in. So initially, we might not see anything, but we want to see after a while what happens to how they look, what happens to how fat they are, what happens to maybe the color. And so this is our initial setup that you actually drew your diagrams of already. And I will bring you the data and the explanation after we finish collecting data for our final masses.